Now it's time to introduce our speaker, Dr. Kristina Cammen. I won't say a lot about the research she's doing because she's going to talk about it herself, but I will say that if you go look at her website, for example, cammenlab.org, it's one of my favorite websites I've seen in a long time of a researcher, because it's both dives deep into the kind of things she and her lab do, her students and colleagues, which at least in terms of her own training, has a lot to do with molecular and analytical approaches to better understand various aspects of the ecology, evolution, and conservation of all sorts of organisms, but particularly marine mammals in coastal systems. She's a leader in an innovative program where there are many important leaders at the University of Maine in this thing called One Health, which is about plant health, animal health, human health, ecosystem health. So she's playing lots of roles there. One of the things you'll see, well, you'll hear it today, but you'll also see it at her lab website, is this deep commitment to training the next generation. She and her colleague Ann Lichtenwallner led a big NSF grant for, I guess, 3 or 4 years called Research Experiences for Undergraduates, which brought students from around the country here to do this kind of One Health research. And I happened to be in the room, I don't know, a month or two ago to hear those students speak, and it was really special. I took notes and learned a lot. But in the course of kind of keeping an eye on Kristina, just because I find her work fascinating, I was looking at one of her papers, the title of which is Predator Recovery, Shifting Baselines and the Adaptive Management Challenges They Create, which I think you'll hear a little bit about today. And one of the lines in this paper really caught my eye, because she was sort of saying, well, you know, we know this part and something might happen in the future that we got to pay attention to, and it's all kind of complicated. And, quote, to address these gaps will require a diverse cadre of natural and social scientists, as well as stakeholders, to tackle the questions of how we define recovery at the species or ecosystem level, and when we have reached this point, essentially, what to do next. And I've got to say that many of the faculty at the Mitchell Center and across this university and elsewhere in the world, regardless of exactly what we've been trained in, and we're all trained in something different, we usually reach a point, if we're dealing with real world problems, where we're kind of saying to ourselves, we're going to need some other kind of help here. We're going to need other academic expertise. We're going to need the help from stakeholders who have lots of knowledge and know-how themselves, right? And we're going to have to piece that all together and try to think about where do we go from here. And so I'm always glad when I see other people who are trying to figure this out, especially at an early stage in their career. It took me a while to learn anything about this. I kind of crashed into it, but she's on the front lines of this in a way that's going to make a real difference, not just for her work, but for her to serve as a role model for many of us about how we can create a better future in the world. So Kristina, thank you for being here.
Thank you so much, David. Welcome, everybody. Those of you here in the room and those of you joining us remotely. It is my pleasure to be here this afternoon and get to be part of this Mitchell Center for Sustainability Solutions seminar series. My goal for the talk today is to share with you a bit of my own story, my training, that moment in my career when I found myself out of my depth and needing other things beyond what I had been trained to do, and the importance of this moment in time for my research career and hopefully my contributions to marine mammal conservation. There we go. So during this seminar, I'm going to invite you to join me on a journey back through time, actually 2 journeys, that of the seal and my own journey. But for our seal, we will journey through time, think about the time in our history on the coast of Maine when we were inhabited, our coastlines were inhabited by the Wabanaki people, through the arrival of the early European explorers, the subsequent exploitation of our marine resources, and then conservation measures put in place leading to recovery. On a shorter, more recent time scale, I will invite you to learn a little bit about myself. I was born here on the coast of Maine, and how I've grown from this small child you see here with a very early love for the oceans and seals into the person that I am today, with still that strong passion and love for the oceans, but perhaps a bit more understanding of the complexity that comes with our coastal ocean environments and stewarding those environments into the future. So I'll begin with my own story. They said I was born in Booth Bay Harbor, Maine, beautiful spot in mid-coast Maine, and I attribute much of my love of the oceans today to memories, or at least stories of memories of idyllic scenes like this, where I would go out on a boat with my family and see seals popping their heads up as we traveled around. Moved away from Maine when I was 5. We translocated to Maryland, to a suburb of Washington, D.C., and I stayed in Maryland through much of the rest of my upbringing. I attended the University of Maryland, where I completed a bachelor's of science in biology and psychology. And from that landlocked institution, which sadly did not have a marine sciences program like we have here, I had to take every opportunity off campus that I could to get my feet wet, both literally and figuratively, in marine mammal science. And one of those awesome opportunities was an internship at the National Aquarium in Baltimore, where I got to help rehabilitate harbor seals like this one that had stranded on the coast of Maryland. And you will see this thread of rehabilitation and seal health that has followed me since that time. I jumped across the pond for my the first part of my graduate degree, completed a master's in zoology at the University of Cambridge. And this was where I got to merge a training and growing love for genetics and lab work that I had gained as an undergraduate in Maryland with my longstanding love for the oceans. And I studied the immune system of gray seals using a genetic approach. Brought that back for my PhD here in the US at the Duke University Marine Lab in North Carolina, where I took those genetic tools and learned some new genomic tools, applying them to understanding aspects of bottlenose dolphin health in the Gulf of Mexico. And then my story comes full circle. In 2014, I moved back to Maine. And I began building connections both here at the university with other faculty and students, as well as with groups like Marine Mammals of Maine, which is a local nonprofit organization that is the boots on the ground or the sneakers on the beach. They're the people who go out and respond to stranded seals and other marine mammals that wash up on our shores in need of assistance. And this is where I'm going to pause my story for a minute. You've seen my molecular training, and you're seeing the beginning of my time here at the University of Maine. We're going to transition to our seal story now, but you'll see me pop in and out of this story as we go through. Okay, so now I will invite you to join me in the Gulf of Maine, and hope that you have all had an opportunity to stand on the beautiful coast of the Gulf of Maine at some point in your lifetime. I'm going to ask you to join me at that spot. Close your eyes for just a moment and imagine that you are standing at your favorite spot in the Gulf of Maine coastline, and think of what you see. Okay. So let me bring us back out of our shared reverie there. And let me ask, what did you see when you stood on that favorite spot on the coast of Maine? For those of you in the room, please go ahead and shout out answers. For those of you joining online, if you'd like to contribute in the chat, I can see that as well. So what did you see on the coast of Maine?
Bar Harbor.
Bar Harbor.
Drifting Beach in Port Clyde.
A beach in Port Clyde. Beautiful. What did you see when you stood at that spot, when you looked out over the ocean or back at the coastline?
So I saw a gray seal come clear out of the water in the midst of a Kobe Menhaden school down not far from here.
Beautiful. So David has just described, for those of you online who might not be able to hear his story, a gray seal coming clear out of the water in the midst of a pogie aggregation, a fish aggregation. What else?
I saw a cormorant like in the water.
Okay, so we have a cormorant in the water, we have a right whale coming out of the fog. So we have some animals in our ecosystem. That's great.
Lots of rocks.
Lots of rocks, yes, physical habitat. And a beautiful description of the Narrows on Mount Desert Island. Fishing boats, there are people in these environments, absolutely. And lobster pots. Great, thank you. So now I'm going to ask you one more time to join me in an imaginary journey. Put yourself back on that spot, close your eyes, and this time I want you to imagine that you were standing on that spot, the exact same spot 500 years ago. All right. And I would ask you what you saw, but my guess is that that vision is a bit more foggy, that we aren't as clear on what our world looked like 500 years ago. I asked that question from the perspective of the seals. I'm curious in understanding how our seal populations in the Gulf of Maine have changed over time, because I think that historical perspective can help us understand how we should move forward. So let me introduce you briefly to our two subjects of today's story. This is the gray seal and the harbor seal. These are two species that exist sympatrically, overlapping in geographic space here in the Northeast US, but also north of us in the Canadian Maritimes, and actually east of us as well in Northern Europe. So they exist across the North Atlantic. And they're similar in many ways, but you'll see they have some important differences as well. So to answer this question, how have seal populations in the Gulf of Maine changed over time? I attempted to answer this question in sort of two different approaches. I first dove into the literature, literally walking into the stacks of Fogler Library to try to understand what I could learn from what had been done or written about in the past. And I also went into the lab and worked with students and conducted research in this area myself. So let me share with you first what I learned about from that literature dive, from those library wanderings, and share with you a brief history of seal populations in the Northwest Atlantic. I'm going to try to cover the last several thousand years of seal history in just the next few slides. So several thousand years ago, our coastline looked very different. This is one artist's rendering of what that coastline might have looked like. It's an artist's reconstruction of a Wabanaki encampment on the coast of Maine approximately 3,000 years ago. And let me take this moment to acknowledge that the land that I am standing on here at the Mitchell Center is in the homeland of the Penobscot Nation. Now in this image, let me draw your attention to our study species. There it is. There's the seal. From images like this, from oral histories, from reconstructions of what a Wabanaki food year might have looked like, where you can see that the seal was harvested by these people, particularly in the early summer months, which we know coincides with the harbor seal breeding season when they are close to shore and hauled out, perhaps for easy access. We know that seals were abundant enough to be harvested by the Native American people. And we know that from archaeological middens that this affected both the gray and harbor seals here. So on this map, this is a summary of the number of artifacts or specimens that have been collected from different archaeological middens across North America. See Maine up here in the right-hand corner, where we have about equal representation of gray and harbor seals in these middens. And then on the West Coast, just to zoom out for a second, you can see that this Native American use, subsistence-based use of these pinniped species is broad spread and has affected our seals, sea lions, and fur seals for several thousand years. Now I'm going to zoom us ahead in time, The arrival of the early European explorers who arrived on our coastlines in the late 1500s and early 1600s. And let me start again with an image. This time it's a map of the region as they might have seen it. And there's our study species, a little bit harder to see in this image, but there's our picture of a seal. And here we also have written records that tell us descriptions of what people saw when they arrived. Captain Richard Strong describes it seeing exceeding great stores of seals. Samuel de Champlain describes stores completely covered with seals, whereof we took as many as we wished. And so one of the trends that I wanted to follow as I traced gray seal and harbor seal history back over time was thinking about abundance. How many seals were there? We don't have numbers from this time of Native American hunting and early European exploitation. We know that they were common, and that's our baseline that we have to work with. Zooming ahead to our next chapter in time, this is a chapter that was quite tumultuous for our harbor seals and gray seals here in the Gulf of Maine, where there were periods of time that swung dramatically back and forth between times when local laws were put in place to protect these seals that were considered attractive to residents and tourists, to other periods of time where there were bounties put in place, where local governments would pay per snout or per head of seal that was brought in, motivating people to go out and kill these animals to remove them from the population. These bounties were motivated by the perception that seals were a problem. And again, we have written record here. I'm going to take us now into the Maine state legislature history. We see records here of seals being called the biggest detriment to the fishing industry of anything that fishermen had to contend with at the time, that seals were killing thousands and thousands of dollars worth of fish, perhaps running into the millions, destroying fish weirs, letting fish out, and fishermen are absolutely baffled about what to do about it. Our fishing industry in the state of Maine has a serious problem. Now, from this period of time, we do have some numbers because, as I said, local governments were paying people per snout or per head. And so they kept records of how many bounties they paid. These are data that were compiled by Lely and his co-authors, showing data for Maine and Massachusetts from the late 1800s to the mid 1900s. And you can see if we culminate, put all this data together, that we reach an estimate that something like 100,000 seals were removed from Maine and Massachusetts during this period of bounties. And again, bounties are not unique to our seal populations here in Maine. You can see there highlighted the bounties that would have affected our populations in Maine and Massachusetts, as I just described, as well as in the Canadian Maritimes. But I will also point out that these bounties are commonplace or have been commonplace in our history for pinnipeds around the world. Many of these similarly motivated by the perception that these seals compete with commercial and recreational fisheries. Okay, so let me take us further one step. And this brings us to 1972, which was the year in which the United States passed federal legislation protecting all marine mammal species in the US waters. This act made it illegal to harass, hunt, capture, or kill any marine mammal species. And we can see the outcome. So if we look now over this period, over the last 150 years, and we begin to think about some numbers now of gray seal, or excuse me, of harbor seal populations, 1947 is the earliest number that I can find. This was the first population assessment for harbor seals in the state of Maine. It was commissioned by the state, and it was actually conducted by a graduate student here at the University of Maine. They counted something like, or estimated something like 3,000 harbor seals at the time. In 1973, after the passing of the Marine Mammal Protection Act, which made the US government federally responsible for monitoring marine mammal populations in US waters, there was the first statewide census in Maine that estimated almost 6,000 harbor seals. And then the early 1980s, NOAA began flying regular aerial surveys, counting seals from plane and using correction factors that allowed us to get a more accurate estimates, knowing something about how much time in a seal's day they spend in and out of the water. And that first estimate came in at 10,500 in the early 1980s, and we can see the numbers then graphed from there. We've steadily increased the harbor seal numbers in the US waters and leveled off seemingly at around 60,000 individuals. So a period of recovery. Here are the data for gray seals. We see exponential rapid growth for gray seals at colonies across the Northwest Atlantic. On the left-hand side of your screen, you see the numbers, population trajectories for pup production at Sable Island, which is an island off the coast of Nova Scotia. This is the world's largest gray seal breeding colony. And you can see its dramatic rise from almost zero in the early 1970s to today. And I will jump forward and say that today, the estimate for the Canadian harbor seal population numbers over 400,000. The panel on the right looks quite similar at first glance. These are the numbers for Muskegon Island, Massachusetts, an island off of Cape Cod, which is one of the largest gray seal colonies in US waters. And again, you see this exponential rapid rise, but notice the x-axis here starts in 1990. Gray seals have been extirpated in US waters as a result of those bounties and were largely absent until the start of their recovery in the 1990s, and we consider them sort of absent from modern memory. There are still people today living on Cape Cod who remember growing up in a time when there were no gray seals. And now, as you will see, they are faced with large numbers of gray seals, and that is sort of our teaser to some of the conflict that might come. In the US, our numbers are currently at about 27,000 gray seals, but gray seals do move and immigrate regularly from Canada. So I put these two numbers up here because the populations are largely linked. And I like to say that gray seals are now so abundant on Cape Cod, they are even visible from outer space. So this is an image from a Google Earth satellite where every single one of those little black rice grains on this beach is a gray seal. And there are actually researchers who've attempted to make population estimates based on satellite imagery like this. Again, zooming out just for a second, I've shared with you the population numbers for harbor seals in Maine and gray seals in Maine, or excuse me, I should say in New England. But this trend of population recovery can be tracked for all pinniped species that breed in the contiguous US following the passing of the Marine Mammal Protection Act. So in fact, this act has been a success story in many ways and has led to the recovery or growth of most of our pinniped species, with some important exceptions in Alaska and Hawaii, where we still have endangered pinniped species in the US. Okay, so hopefully now we have some sense of an answer to this question. How have gray seal and harbor seal populations changed in the Gulf of Maine over time? But I mentioned that I took two approaches to answering this question. One was to go into the literature and look at data. The other is to use my lab-based skills, molecular training. And here I'm curious to understand how the genetic diversity of these seals has changed over time, recognizing that genetic diversity can tell us something about population size, population history, and the fitness and adaptive capacity of populations today and forward looking into the future. All right, so to answer this question, I collected a bank of tissue samples and brought those into the lab. And I collected those samples from across the Northwest Atlantic. I had samples from gray seals that were found in the Gulf of St. Lawrence, or breeding in the Gulf of St. Lawrence and Sable Island, that large gray seal breeding colony off the coast of Nova Scotia, and on Muskegon Island in Massachusetts. And I also had samples from harbor seals that were collected from a Northeast Fisheries Observer program, so from animals that were bycaught in fishing gear. And this is one of the spots in our journey where I'm going to interject myself back in just momentarily. As I mentioned, I'm a molecular ecologist by training. The lab is my happy space. I also love getting out in the field, but I am not a field biologist. And so to conduct research like I'm going to describe to you now, I rely on large collaborations. For example, the wonderful people depicted in this photo who represent individuals from federal agencies, from other academic institutions, and from local nonprofit organizations like I Found Marine Mammals of Maine. This is the team that goes out to Massachusetts beaches every winter, every January, and conducts the live capture health assessments of gray seals that end up producing the seal tissue samples that come into my lab. So let me just show you what that looks like, even though it's not my story on my own to tell. This is what a beach of Massachusetts looks like. Happens to be January 7th, 2016. This is gray seal pupping season. Gray seals give birth to their pups in the middle of our New England winters. And so the darker blobs that you see here are their gray seal moms next to the smaller, whiter blobs, which are our pups very early in the breeding season. As the pups get a little bit older, their coats start to darken. And when they are weaned and no longer nursing, then they are ready to be sampled. And so here are two of the collaborators who are collecting a gray seal pup, bringing it back to the sampling station. They weigh the pup. They take blood samples for disease testing, biological samples for multiple different types of analysis and diagnostic work. And those are the, they put flipper tags in these animals, so similar to a cattle ear tag, so that the animal can be identified if it is resighted. And it's this flipper tag, essentially like the piercing of an ear, that results in the teeny tiny skin tissue plug that comes back to my lab and then produces the genetic data I'll show you momentarily. And then because I couldn't help myself, here's one more photo. Okay, so that's how I get some of my tissue samples for this genetic analysis that represent the animals who are alive today. And I can understand something about what those populations looked like in the past, just from looking at the genomes and the genetic diversity in the animals that are alive today. But it is also helpful if I can get some samples from those animals that were alive, say 500 or 600 years ago. And to do that, I have leveraged collaborations with the University of Maine Archaeology program and the Passamaquoddy tribe to get artifacts from the Machias Bay Archaeology Field Camp. And so this is work that I was able to do in collaboration with the late Dr. Brian Robinson and his graduate students, including Sky Heller. I'd like to acknowledge again here that these sites are on the homeland of the Passamaquoddy, and the study of the artifacts that are collected from these sites have been carried out for many years in close collaboration with Brian Robinson and his students, who have consulted closely with the Passamaquoddy Tribal Historic Preservation Society. So from those either skin samples or bone samples that I bring back into the lab, extract DNA from, I then amplify regions that can tell me something about their genetic diversity. And I can map out that genetic diversity in networks like this. Here, each circle on this figure represents a different genetic variant. The size of that circle represents how common that variant is in the population. And you can see that some of these are pie charts representing different frequencies of that variant across the 3 gray seal populations in this case. And then you can see that the circles here are connected by lines in between them, and the distance between circles represents the genetic distance between 2 different variants. So these are the data from our modern or contemporary gray seals, and what you can see here is a diverse network dealing with a genetically diverse large population of seals. I can compare this to the network that I similarly create from the archaeological specimens to get a sense of how the now compares to the then. And so that archaeological network is up here on the top of the slide. And what you'll notice here, perhaps if you can see it, is that there are a bunch of small white circles. These are gaps. These are variants that are not present at that time point. You might notice as well is that our archaeological network has far fewer colored in dots. I had far fewer samples. We're talking 8 samples in the archaeological network as compared to almost 400 in the modern day network. But what's interesting to me here is that of those small number of samples from the archaeological specimens, they were all distinct. They were all different genetic variants, suggesting that they were pulled from a large and genetically diverse population. I've connected for you with vertical dashed lines here 2 variants that were the same. Well, 1 variant that was found, I should say, in both time points. There was only 1 variant found in both time points, suggesting that that large diverse population of gray seals that existed in history might have looked a little bit different than the gray seals that are here today. And here's the data for the harbor seals. Again, you can see diverse populations, but what stands out to me at first glance is that we have more vertical dashed lines. There's more similarity between the harbor seal population back then and the harbor seal population today. And so from this initial genetic analysis, I draw this conclusion that there are large differences between the seals that I sampled in modern day time and from the archaeological specimens, and that this difference is perhaps even greater so for gray seals. I can also test specific hypotheses. So I mentioned that even in just the genomes from the contemporary samples, we can look backwards in time because our genomes have signatures of what has happened in the past to our ancestors and those to which we had arisen. And so here I test 5 specific hypotheses or sketch them out as changes in population size from past on the top of these schematics to present at the bottom. You can see a scenario of no change in population size, an expansion scenario where maybe the population got bigger over time, a reduction scenario, maybe it got smaller over time. Getting more complicated as we move from left to right. We have a bottleneck scenario where the population got small and then got big again. And maybe we combine all these together. Maybe first it expanded and then it got small and then it got big again. You can imagine the iterations that one could go through, but I stopped here at 5. And our genetic data from those contemporary genomes strongly support either of these 2 population bottlenecks, suggesting that that history that I described to you of exploitation followed by recovery, we see that the lingering impacts or lasting impacts of that history in the genetic diversity of the seals that are alive today. And we can model back in time what our population sizes might have looked like. And you can see here in these 2 models, population size on our y-axis, and then from left to right, we're now going backwards in time as we go to the right. So particularly for our gray seal, and maybe not quite so much for our harbor seal, we see that our gray seal population might have been larger than it is today. So if we think about the implications for this type of genetic data for protected species management, particularly in a system that lacks those historical baselines, we know that seals used to be common, but how common is common, we don't really know. We can get a little bit more information from that or related to that question in these genetic data, which provide evidence of large historical populations as well as lasting impacts of those population bottlenecks. Okay, so we've traced this history of seals from our time of Native American hunting, early European exploration and exploitation, depletion of the populations, protections put in place, and recovery of our numbers. And then let's see what happens next. So our next chapter here, Beyond Recovery, begins to bring in some of the issues that I don't have all the answers to as a molecular ecologist, and where I begin to find myself out of our depth. And I'm going to start by drawing a connection here between the gray wolf and the gray seal. And this connection is found both in the translation of gray seal into French, where it's actually called the marine wolf. But it's also there in its history. So both the gray wolf and the gray seal were exploited. They were hunted. Their numbers plummeted. Conservation measures were put into place. Their numbers started to rise. In the case of the gray wolf, some of this was through intentional reintroductions. As those numbers rise, there are sectors of our public that celebrate that conservation success. And there are sectors of our public that freak out, because we have predators that are now back in our backyards. And how do we handle that? Remembering, right, that many people grew up without ever seeing a gray wolf or a gray seal in their backyard. And they're quite used to that happy sort of baseline. For seals in particular, the concern with their recovery comes from a few different roles that they play in our marine ecosystems. The first is their role as predators. As you might have guessed from those bounty programs, there is still lingering concern over the role that pinnipeds play as competitors with our commercial and recreational fisheries. There is new concern over the role that pinnipeds play as prey. So this image shows us a group of gray seals in the bottom left-hand corner, basically waiting to swim the gauntlet past that great white at the top right-hand corner of this image. And there's concern that seals are part of the reason that we are seeing more and more sharks in our waters here in New England. And then there's concern over the roles that gray seals in particular play as competitors and as disease reservoirs, and the potential threat that might spill over to the more susceptible and less numerous harbor seal as one population rises and the other begins to level off. And I'll come back to that disease component in just a bit. So these are some of the issues that, as I said, I can't address on my own as a molecular ecologist. Genetics can only take me so far. And so I have, over the past several years, as I've found myself out of my depth, continued to expand my horizons, working in particular with other colleagues who can help me conduct interdisciplinary science for marine mammal conservation, and learning more and more from stakeholders who have different perspectives on these issues. And at the University of Maine, I've been fortunate to do this as part of the Mitchell Center for Sustainability Solutions, as part of the One Health initiative that David mentioned earlier, that ties together animal health, human health, and environmental health. And more recently, as a member of the Maine Center for Genetics and the Environment, that's really trying to bring together these genetic tools and environmental science. And the stakeholders that I've been fortunate enough to engage with, some of them are depicted here on the left. As I mentioned, Marine Mammals of Maine, that member of the Marine Mammal Stranding Organization, a nonprofit group that works in the southern half of our state, the Maine Department of Marine Resources, and NOAA Fisheries. One of my first forays into this space is that paper that David mentioned. This was a paper that I published just towards the end of my postdoc, and I still call it the hardest paper that I have ever published, in that it took multiple iterations to get it right. And that's because I was doing something I wasn't used to doing. I had at this point learned how to write a scientific paper about genetic data. I didn't know how to write one that didn't have genetic data in it. And I worked here with Doug Rasher and Bob Stenek, who have a bit more experience in this space. I'll tell you a little bit about what came from that paper and from that process. So the first are the two figures I've actually already shown you in this talk. The first thing I did in writing this paper was seek data. And so in this case, I sought did a literature review. I found the data from the archaeological middens, and I found population numbers that have been taken from these census of different pinniped populations across the US. But the review of this data wasn't the hard part for me. As a scientist, I like data. I work with data. I can speak data talk. But what being part of the Mitchell Center and other interdisciplinary stakeholder engaged collaborations has pushed me to do is to think beyond that data and to think about the policy and management implications. And that is definitely where I find myself out of my depth. So here what we did is we mapped out in time this sort of cycle or this paradigm that we observed of population decline, followed by conservation measures put into place, followed by population growth and the start or achievement perhaps of recovery. And then the question becomes, what's next? And we said, okay, well, maybe it'll take one of three paths. We have the status quo, which is sort of what the Marine Mammal Protection Act is for us right now, where we maintain protections in place for these marine mammal species, regardless of their conservation status. We have reduced protection, like maybe the delisting of a species under the Endangered Species Act, or maybe we have a policy change, and this is something that is increasingly being called for, that we revisit the Marine Mammal Protection Act and see if it still fits our current day paradigm, or should we be relaxing additional protections? But if we do that, do we just start that cycle anew? I don't have answers to those questions yet. I'm not sure I'll get there. But what I can do is think about these different potential impacts of pinnipeds as they recover in our ecosystems. And I can take my molecular tools and my new collaborations and my new partnerships and attempt to study the impacts of marine mammal recovery to help us inform the decisions that we have to make coming forward. So I want to spend just a little bit of time sort of foreshadowing my future. And to do that, I have to bring in my students, who are a huge part of the research that I do now. And this is just a few of them, but I'm going to introduce to you a bit of the work of Julia Sinnerberg, who's a PhD candidate in the Maine eDNA program, Kristina McCosker, who's a PhD candidate in the One Health and our graduate NRT program, and Amanda Cruz, who was standing up here a month and a half ago presenting out on her REU experience she spent this summer with us. And just a quick plug. I can only share so much today in my limited time about what Julia and Kristina are up to, but come learn more and hear directly from them at a School of Marine Sciences seminar, Friday, October 28th at 11 a.m. And I can share with you information about how to connect to that or find that seminar if you're interested. Okay, so Julia's work is thinking about how can we develop eDNA or environmental DNA approaches for non-invasive sampling of marine mammals and their communities. eDNA leverages this idea that we all leave traces of ourselves, traces of our DNA behind us in the environments in which we live. And so by collecting a water sample, we can extract the DNA that's free floating in that water sample and learn something about the animals that are present or were present in that body of water in some near past, without ever touching that animal, without ever seeing that animal in many cases. This is exciting. It's got a lot of potential for non-invasive sampling and broad ecologically minded sampling for marine mammal species. We first trialed this work in a controlled setting, again, leveraging those partnerships with rehab organizations. Remember that college undergrad version of myself working in a similar rehab setting. This is Sarah Burton. She was an undergraduate student in our lab for several years. And here she is collecting a water sample from a pool of water that housed a rehab seal. And we were able to take that water back and make sure we could actually get that seal's DNA just from that water, which we could. We then have gone out in the field, and here's an image of Julia out sampling the Cape Cod gray seal haulouts. So those haulouts that I described to you earlier, there are these intense health assessments that occur on these beaches, and we get tissue samples from those animals. But can we also get their DNA just by scooping up a liter of water offshore? And this is work that's been funded in part by Maine eDNA and conducted in collaboration with the Center for Coastal Studies. And Julia's work so far is showing us that, yes, we can detect gray seal eDNA in these water samples consistently 50 meters from shore, and in some cases even up to 150 meters from shore. And yes, as expected, there are higher concentrations closer to shore, but we might be able to push that envelope and really collect these samples at a distance that have minimal disturbance to the animals. We've also pushed the envelope in the genetic data that we can collect from these samples, so we can not just detect gray seal eDNA, but we can also characterize those genetic variants, and that perhaps even begin to make some of those networks that I was showing you earlier that were made from tissue samples. And then we brought this sampling approach closer to home. A sampling with Justin Stevens from Maine Sea Grant out on the Penobscot River, work funded by the Maine Outdoor Heritage Fund, where we are looking to see how can we co-detect and perhaps describe the spatial distribution of both gray seals and their potential prey, like the river herring, from which you see some data here early of the summer last year. So then switching gears to Kristina's work, I mentioned that one of the concerns for gray seal recovery in New England is that they might play a role as a disease reservoir, and that as this population grows, we might have a growing number of animals that are capable of circulating diseases that can spill over and have larger term impacts on our more susceptible sympatric, for example, harbor seal. Harbor seals have been the bearer of several recent mortality events, including outbreaks of both bovine distemper virus, which is a measles type virus, and influenza A virus. And then this most recent summer, 2022, there was an outbreak of the highly pathogenic avian influenza that seems to be spilling over from our bird populations into our harbor seal populations, but which we think can circulate with limited impact in terms of negative health impacts or outcomes in our gray seals. We're attempting to address this question of why are gray seals more resistant and what might the potential impacts of recovery of gray seals mean for disease dynamics in the system in sort of a tripartite approach here, working in the lab, in the field, and in practice. In the lab, we're asking questions like, are there differences in immune system genes between gray and harbor seals that might explain why one species is more susceptible than the other? Within gray seals themselves at different infection stages, we're trying to understand the genomic factors that influence the progression of disease and their resistance to disease. And then from those harbor seal outbreaks, we want to know if there are differences between the harbor seals that do and do not survive when exposed to disease. In the field, we're collaborating with those marine mammal stranding organizations, leveraging the awesome long-term data sets that they are collecting from animals that strand on our beaches, summarizing when, where, and why marine mammals strand in the Gulf of Maine, and in particular, considering them as sentinels of ocean health. By studying the marine mammals, what can we learn about the broader ecosystems or the health of the broader ecosystems in which they live? And so we can track numbers of strandings over time. We can see peaks during those outbreaks. The blue line here is our gray seals, and we can even see that rise in our gray seal population reflected in the rise in the number of strandings that we see in our state. And we can map these out and identify hotspots of strandings or even hotspots of human interaction, and think about mitigation and public outreach programs to help address some of those hotspots. And finally, in practice, we can work with other stakeholders in the region. So I'm a member of the Northwest Atlantic Seal Research Consortium. It's a multi-stakeholder group that really takes this ecosystem approach and considers healthy marine ecosystems, those that include healthy populations of all marine resources, from fish to seals to whales to sharks. And we can begin new collaborations like Kristina's upcoming internship with the Marine Mammal Commission, where she will help contribute to building web-based data management platforms that again try to link those connections between oceanography, ecosystem processes, and marine mammal health through the Marine Mammal Health Map. So this has been Gulf of Maine focused. But again, as I've zoomed out a few times in our review of data, let me zoom out just one last time to highlight some emerging work. Melissa Fly is a PhD student here in EES. She is not one of my advisees, but I am fortunate to be one of her committee members. She's co-advised by Carly Spinarski and Joseph Lusky, and she's working closely with NOAA Fisheries as they attempt to understand across NOAA offices in the United States, how do these federal managers and scientists view this problem of marine mammal recovery? What are the human wildlife conflicts that they are observing, and how can we help them chart their future? So this brings me near the end of our journeys through time. Thank you for following my journey for our gray and harbor seals from the time of Native American hunting through today, for following my own journey from that small wide-eyed child on the coast of Maine to the assistant professor that I am today, and hearing a bit about how I've found myself out of my depths. There was moments in my time and my career where I've said, I don't have all the skills. I need to find the other people who have those and work with them. And those collaborations have been immensely enjoyable and productive. And I'd just like to conclude by sharing some of my inspiration, which comes from some folks in my close vicinity who I watch finding themselves out of their depth on a regular basis. This is what my life looked like this weekend, from Common Ground Fair to soccer practice to apple orchard picking. Let me introduce you to Willa, my 6-year-old daughter, and Brooks, my 1.5-year-old son. As a parent, I find myself continuously out of my depth, trying to help these two navigate the crazy world that we live in. But I am inspired on a daily basis as I watch them push themselves outside their comfort zone from minute to minute, trying new things and seeing what happens and maybe repeating it or maybe not, depending on what kind of reaction it gets from the rest of us in the room. And I take this moment to encourage all of us to push ourselves out of our depth, out of our comfort zones. And it might be scary, but we can see the productive successes that it can lead to, from professional to personal. Tons of people to acknowledge. I can't list them all, but here are some of the folks who contributed the biological samples that I said I don't collect myself as a non-field biologist, the many funding sources that have contributed to this work, the collaborators and the students that have helped me get here. And thank you all for your attention and attending today. I'm happy to take any questions. Thank you.
Oh, let's give her a hand. That was just, oh my goodness, that was so wonderful. That was really, really interesting. I imagine that every single person who's here today is already thinking 2 or 3 things. Who am I going to share this with? How can I be involved? And I've tried to capture some of what I think people, maybe thinking about here. And we'll start asking some of those questions if you don't mind. So the first thing is, we can really see the importance of your work. We can really feel the importance, and also the beauty of having you say, I don't have all the skills. And also hearing you talk about being born on the coast of Maine in Booth Bay, and then moving to Maryland, and then coming back and having 2 separate interests: love of genetics and love of the ocean. When did you start to see those coming together?
Yeah, I think you've captured it really well there, Linda. I do. I love being in the lab, but if I was in the lab every day, all day, I would miss the ocean. And when I'm on the ocean, I sometimes miss the control and organization of my lab environment. That's me. I think I saw them begin to come together as an undergraduate. It was my first experience. Well, actually, it was my second. My first experience was working with fish in tanks. I didn't like that so much. But my second research experience as an undergrad was in a genetics lab. So I learned some of the skills there and was working on oysters. Didn't feel passionate about the oyster-related research questions, but began to see the value of these skills and then combined them in graduate school. So took these new skills and the animals and questions that I was passionate about, the questions related to ocean conservation and marine mammal health, and figured out how to bridge those 2 otherwise pretty disparate fields.
Really very interesting. It sounds like you had growing capacity to solve problems and to see the importance of bringing together many kinds of knowledge. Were there ways that you told people about that, the importance of bringing together different kinds of knowledge, and did they hear you?
I think I've been able to maybe share that message most effectively with my students. I think I learned it in graduate school when I was surrounded by peers who were studying many different things from the natural to social sciences, and I saw value in all of those different aspects of studying the oceans and the policy that governs them. And so I have sort of shared that experience and my story with others in sort of the way that I did today. I think it resonates a lot with students who are coming up in the field of ocean conservation. And so that has been maybe the most friendly audience that I have seen for that message. So it doesn't mean that other folks later in their careers can't come around to it too.
Yeah, very.
David shared in his story.
You did something really interesting near the beginning of your talk. You asked us as an audience to think about our own experience. You said, what do you see on the coast of Maine, and asked people to respond to that. And then you said or asked, well, 500 years ago, what would it have been like? You asked us to sort of envision a historical perspective. That's really helpful in a lot of ways for getting us to think, longer term. Do you find that people respond well to that? Does that get them ready to kind of think about these issues? Or what are your thoughts about that?
Yeah, I've used that approach in many talks that I've given, and I love the responses that I get. Because I think it does two things. One, it shows us how different our views of the world might be, that one person looking out might see seals while the other person looking at the exact same spot of water might see boats and lobster pots. And we have all of those views or world views to contend with. And then I do think it helps people maybe appreciate pretty acutely the challenge that we have of knowing what was here before. And yet it's something that I think many of us acknowledge is important as we think about what we want the future to look like. I mean, from grade school, right, we learn history because history is we're not supposed to repeat history's mistakes, just to learn from history as we think about how to move forward. And that's still what I'm trying to do here.
Yeah, one of the really interesting, thank you very much. You talked about the importance of using various written records and also using sort of archaeological information. Have you found, as you think about the different kinds of records or kinds of information, that that's another area where you start to see the importance of disciplines coming together?
Yeah, it's been fascinating. And that's an area I'm still learning about. I think the different ways of knowing the world is something I'm still learning to appreciate the multiple different ways of knowing the world, the different types of data that we have. But I do see a lot of value in the diversity that is available to us. And I will say that came about because came here as a postdoc talking about genetics and seals, and Sky, that graduate student, said, we have some artifacts of seals. Are those useful to you? And it opened my eyes to this whole new set of data and new collaborators and new ways of knowing in a really exciting way.
You did, what a great point. And you even, one of the very interesting things you said as you were talking about using different kinds of information, you talked about things that one might collect or see on the coastline, but you also, early on, you said library, you went to the library to find that. So how does thinking about both of those things, does that for you again raise the issues of the importance of different disciplines? And do they draw on somewhat different information sources?
Yeah, I think absolutely. I think in the same way that I couldn't do my genetics work without the people out there collecting the samples in the field, the people understanding the ecology of the animals and the oceanography of the environment, I don't think we can understand our history without these different types of data, without the library stacks and the oral histories and the written histories and the images, piecing it all together. I think I'm just starting that process.
And people in the chat are thanking you. Like one of the people in the audience says, thanks, Kristina, great presentation. Will eDNA studies take the place of radio collars and other physical tracking systems to help map populations of marine mammals and other species in the future?
I think eDNA has a lot of potential. I don't know that it's going to replace anything. I think we're going to use it in complement. I haven't thought specifically about tagging and tracking, but I have thought a lot about physical sampling of animals, and I think, and I should say physical sampling of animals or monitoring of populations. And there, I think it's got different strengths and weaknesses to the other approaches that we have. And so, again, in the same way that bringing together different ways of knowing, we can bring together those different approaches, we might get a richer image. But I think any one approach, including eDNA, is not going to have all the answers.
Right. We all, I think, want to follow you around. We want to be a part of your team. And David may have a few questions related to that people in the audience, not the virtual, but the in the room audience. David, do you have a question or two?
So thanks, Linda. And any questions here? I have one, but if somebody else has one before me, I think you guys should go first. Anybody? Okay, so mine is kind of this distant past and present. So if I understand you correctly, there are people who have been concerned for a long time about there being too many seals and their effects on fish, for example, and fisheries. But am I right? So if particularly for gray seals, it looks like going back a ways, there might have been more gray seals thousands of years ago. But also the kind of things we hear about from both Wabanaki stories and early colonization stories about there being super abundant fisheries here.
Yeah.
At the same time as there might have been more seals. Does anybody have does that cause anybody to wonder how that might have worked?
Yes. So the question is that we have, we think that there used to be a lot of seals. We also think there used to be a lot of fish. And how can those two things coexist? I think our ecosystem had it was in a different stage at that point, and it's shifted so dramatically because of human exploitation that people talk about there used to perhaps have been cod so big that the cod competed with seals instead of seals eating cod. It was just a different system. And so the predator-prey dynamics, some of those were probably the same and some of those might have looked of a different flavor back then.
Yep. So I just looked at the time, and unfortunately we're at time. For people who want to learn more from Kristina, please come up if you're in the room, or connect with her easily. You can go to cammenlab.org and find out all about all of this great work. But for now, let's all thank Kristina for
Yes.